first day of this meeting is devoted to the cost action H Comet. And so I'm going to give a quick overview of the action. A little bit of history. How did H Comet start? Probably about 10 years ago, we launched a project which we called ComNet. And the aim of ComNet was to collect as much as we could of the Comet Assay data from human trials and create a common database, and then to carry out a pooled analysis. And the main aim of ComNet really was to find definitive answers to questions that had been asked by various people and often obtaining differing results. So the only answer was to put all the data together. And these questions were, for example, does DNA damage increase with age? Are there differences in DNA damage between men and women, between smokers and non-smokers? And what are the effects of exposure to various environmental and occupational mutagens? The model for the pooled analysis was the work that had been done over recent decades on other biomarkers used in human biomonitoring, chromosome aberrations and micronuclei. And it had been clearly shown that these biomarkers can predict cancer risk. In other words, if you have a high level of chromosome aberrations or micronuclei, there's a higher risk that you will contract cancer in the future. So there's a further challenging question that we can ask in the Comet Assay field. Is DNA damage also a biomarker of cancer risk? Now, this may seem obvious, but there are certain things to consider. There is evidence that DNA damage can be elevated in cancer patients, but this could easily be an effect of the disease rather than a cause. And DNA damage, of course, is an initiating event in carcinogenesis, but it's worth remembering that almost all of the damage that is incurred is repaired. And so the link with cancer is tenuous. So what we really need is cohort studies where we can follow a large group of individuals measuring DNA damage or other biomarkers, and then over the following decades look for incidence of disease to see whether there is a link between DNA damage and cancer incidence. Well, ComNet was never funded, but in spite of that, we managed to recruit over 100 researchers, half of whom agreed to provide us with their Comet Assay data. On this basis, we applied for a cost action to allow us to carry out the aims of ComNet. And we had a secondary aim, which was to try to standardize the Comet Assay so that there were fewer variations between laboratories and we could carry out comparisons and collaborations more reliably. And this cost action was launched in April 2016 and lasted for 4.5 years. In the end, we had 25 full member countries from really most of Europe. We also had observers from near neighbor countries, Jordan and Kosovo, and from international partner countries, India, Cuba, Brazil, and the USA. So this shows the very wide coverage of Europe with some surprising exceptions. The two green countries are the two near neighbor countries, Kosovo and Jordan. It's worth pointing out that more than half of these member countries were inclusiveness target countries, which is the cost way of referring to countries that are less research intensive and therefore warrant particular attention and assistance. As Sabine said, the cost action was organized around working groups. The first and most important working group was concerned with creating and analyzing the database. Then there was a working group which was looking at improvements in the technical aspects of the Comet assay. We had a ring study to try to sort out variation in Comet assay techniques between labs. A working group was dedicated to devising guidelines and standard operating procedures, part of the attempt to get people to use the same techniques in their experiments. We had a working group on DNA repair, which is, of course, another important aspect of genome stability. We looked at different cell types that are used in human biomonitoring. Usually lymphocytes are the sample cell type, but others can be used. And finally, there was a working group which was planning for the future. So you'll hear about the progress made in some of these working groups in the following presentations. Just in case there's anyone in the audience who is new to the Comet assay, I'm just going to very quickly go through it. Cells are embedded in agarose on a glass slide. They're lysed in detergent and high salt, which removes almost everything except for the DNA, which remains attached as supercoiled loops to a matrix, and the structure is called a nucleoid. There's a period of alkaline incubation, which allows the DNA to unwind a bit and also releases alkali labile sites as breaks. Then there's electrophoresis and neutralization, staining, and fluorescence microscopy. And we see an image like a comet where the DNA in loops that contain a break has relaxed supercoiling and is able to extend towards the anode, forming the tail. So the intensity of the tail reflects the frequency of breaks. There's an optional extra step, digestion with a lesion-specific endonuclease. The standard assay measures strand breaks and alkali labile sites. If we include the enzyme, such as formamidopyrimidine DNA glycosylase, or FPG, this allows us to measure base damage in addition to strand breaks. And in the case of FPG, we're measuring the frequency of oxidized purines. So it adds another level of information to what we can get from the Comet assay. Okay, so as I said, the first working group was concerned with the database, and we received almost 20,000 individual sets of data, variously reporting on strand breaks, enzyme sensitive sites, hydrogen peroxide sensitivity, DNA repair capacity. And these data came from 108 human trials and 38 research groups. As you can imagine, analysis was a mammoth task, and you'll be hearing about the findings that have come from this working group in the next talk. The second aim of H-Comet was Comet assay variability. For many years, we have been concerned with the variability that is seen when you compare results from different laboratories. And this has been revealed in collaborative studies, particularly ESCOD and ECVAC, which took place over, well, the past couple of decades. These differences can arise from technical factors, and it's clear that many researchers have adopted the Comet assay as a simple technique and then have introduced their own variations, which is fine, except that it makes it very difficult to compare results from different labs. And often these modifications are made without a full understanding of the underlying principles of the assay. So in H-Comet, we've emphasized the influence of specific practical aspects of the assay on results, particularly those that are really important. So the agarose concentration is important. The more concentrated the agarose, the more difficult it is for the DNA to migrate towards the anode. Lysis varies in different labs, different projects, can be even less than an hour, or it could be up to days or weeks, and it does have effects on results. Enzyme incubation conditions are important. It's crucial to optimize the digestion conditions for the enzyme. Then the time in alkali can vary, and depending on how long the gels are in alkali, we can get more or less conversion of alkali lamellocytes into breaks. Really crucial is electrophoresis, and the formation of the comet tail depends on the time of electrophoresis and also on the voltage gradient across the gel. And finally, the scoring method has turned out to be one of the main causes of variation between laboratories. So we have been developing standard operating procedures in the hope that they'll be adopted by researchers, and this will reduce the variability and make comparisons between laboratories more reliable and collaboration more straightforward. Although the most commonly used sample cells in human biomonitoring are lymphocytes, so-called, actually, strictly speaking, peripheral blood mononuclear cells, other cell types may be more convenient or informative. For example, cells from the lining of the mouth or even from the surface of the eye can be used in a fairly non-invasive way. However, a lot of our work has been done to look at the possibility of using samples from biobanks. Biobanks have been established during large-scale epidemiological studies, such as EPIC, the European Prospective Investigation of Cancer and Nutrition. And these biobanks have stored frozen whole blood or buffy coat samples. Buffy coat is a red cell sample enriched with white cells. And we found that these samples can yield leukocytes that can be analyzed with the Comet assay even after decades of storage. So that means it's possible to envisage carrying out a nested case control study. In other words, we would identify a group of patients in the cohort and a matched group of controls and go back to the biobank, fish out the samples and measure the DNA damage from maybe 2 decades or more before the disease occurs. And there'll be more about this in a later talk. DNA repair can be measured with a variant of the Comet assay. The graph shows data from about 30 people measuring their repair rate on 2 occasions. You can see there's a correlation between the 2 occasions, meaning that individuals have a characteristic repair rate. And there's a very wide variation between individuals. So what's the significance of this variation? Well, the faster cells repair DNA damage, the less likely it is that the damage will lead to mutation and cancer. But on the other hand, a high repair rate might represent a cellular response to exposure to a damaging agent. So studying DNA repair rate alongside DNA damage in human trials should help us to resolve these issues. Apart from the scientific questions, we were very keen to carry out training. An important objective of HCOMET was to create a cohort of skilled young molecular epidemiologists by providing training in the form of courses and exchange visits. We had a series of very successful training courses on different topics related to the Comet assay in different cities, and they were, I think, appreciated by those who attended, and they were a very good opportunity to get to know each other and to see various cities in Europe that perhaps wouldn't otherwise have been visited. The short-term scientific missions were an important aspect of H-Comet, and we were able to support 32 of these missions. They provided training for mainly young researchers in particular techniques, and also valuable experience of working in another lab in another country. In addition, they contributed significantly to the scientific output of H-Comet. As I mentioned before, many of our member countries were ITCs, inclusiveness target countries, and they were eligible for special support to attend conferences. And we were able to support 26 young scientists in this way. Our output was really very impressive, I think. We published over 70 papers, including more than 40 with co-authors from 2 or more partner countries. We had a 2-part special issue of mutation research, which was commissioned. 2 papers have appeared in Nature Protocols, and another one is in progress at the moment. Really a spin-off from the working group on guidelines and SOPs, but involving a large number of members of H-Comet. Associated with these papers is a set of training videos, which I think will have a lasting influence on the Comet Assay field. The website is still in operation and will continue to provide access to videos, details of publications, guidelines, SOPs, and so on. And all H-Comet members and others are invited to keep in touch through the International Comet Assay Working Group that Sabine has told you about. So that's my overview of the project. And here are some happy members at one of our meetings. Thank you.